Press the bell icon on the YouTube app and never miss another update.
Zero vidana. Okay. Namaste, children. Yes. Good morning, Kirtana, Rekha, Divya. Good morning. So in this class, we'll discuss cell unit, the unit of life. So what is cell? What can anyone tell me what is cell? You already read in your lower classes. Kirtana, good morning. So when we look around, we see both living and non-living things. So there is a question that the living things, what the living things are made up of, that is not seen in non-living things. So the answer is cell. Cell, what is cell? Anyone? Amsashri, good morning. Cell is considered as structural and functional unit of an organism. Understood? So in this class, we'll discuss about cell and the organelles present inside the cell. Abhishek, good. It's a structural and functional unit of life. Amsashri, good. So when we see this cell, cells are of two types, prokaryotic cells and eukaryotic cells. Some organisms are made up of single cell. Those organisms are called unicellular organisms. Some organisms are made up of many cells. Those are called multicellular organisms. Kirtana, good. Two types. In that you have answered unicellular organ. No, I asked cell. Cell, it is a structural and functional unit of life. So in the prokaryotic cell, even though we have many types of prokaryotic cells with different functions, but the structure remains the same. So in the prokaryotic cell, we have, this is outermost is cell envelope. Inner to that we have cell wall. Then we have cell membrane. So in prokaryotic cells, we see outer is cell envelope. The next to that is cell wall. The next is cell membrane. And they don't have any cellular organelles. So nucleus is not enveloped by nuclear membrane. It is genetic material is nucleo. It is not surrounded by nuclear membrane. Nisha, good morning. And we see membraneless organelle, that is ribosome is present. 70S ribosome is present. And one more important structure is present in prokaryotes, that is plasmid. So it is a circular DNA, extra chromosomal DNA present in prokaryotes. Importance of plasmids we'll discuss in biotechnology when in your second PUC. So, and in prokaryotes we see cilia and flagella, extension of cell membrane. So this is, and one more important characteristic feature of prokaryotic is mesosome. This is mesosome. So infolding of cell membrane mainly helps in cell division in prokaryotic cells. So coming to the eukaryotic cells. In eukaryotic cells, we see different types of cells with a different function. So we all know that plant cell and animal cells. So in plant cell, we see cell wall, large vacuole and chloroplast. So these three are seen in plant cell. Which one? Cell wall, vacuole and chloroplast. Whereas the cell wall, large vacuole and chloroplast are not seen in animal cell. In animal cell, we see small vacuole, cell wall is absent and chloroplast is absent. whereas centriole is present in animal cell, that is not in plant cell. So like that in eukaryotes we have different types of cells. So Robert Hooke first he called cell in an empty hollow space surrounded by wall in a cork. Is a dead cell. So ಅವರು ಏನು ಮಾಡ್ತಾರೆ ಕಾರ್ಕ್ cambium ಸ್ಟಡಿ ಮಾಡುವಾಗ he saw empty cell, empty space surrounded by a wall, he called that as cell. Whereas living cell was studied by Anton van Leeuwenhoek in protozoans. So Robert Hooke gave cell or dead cell that is seen in cork. Anton van Leeuwenhoek he studied living cell, live cell in protozoans. And one more scientist, Rudolf Virchow. So he gave omnicellular. E-cellular means new cells arise from pre-existing cells. Rudolf Virchow gave omnicellular. E-cellular means new cells arise from pre-existing cells. So based on this, cell theory was given by Sheldon and Swan. So they told that all organisms are made up of cells and products of cells, and also new cells arise from pre-existing cells. So that is about cell theory. Now we'll discuss the structure and function of each organelle present in cell. Arsha, good. Omnicellular, E-cellular, given by Rudolf Virchow. Adra meaning ಏನೇಳಿ new cells arise from pre-existing cells. So that's the meaning of omnicellular, E-cellular. So ಇದನ್ನು consider ಮಾಡಿಕೊಂಡು Sheldon and Swan they gave cell theory. So cell theory includes all organisms are made up of cell and components of cells, products of cells, and the new cells arise from pre-existing cells. So coming to the plasma membrane. So in plant cell, outermost cellular organelle is cell wall. It is seen in what's the function of cell wall in plant cells? What's the main function of cell wall in plant cell? Yes, anyone? Protection, protects the cell. Yes, Arshad, boundary of plant cell. Sumuk, avoiding bursting of cell. That's not the answer. It gives definite shape to a cell, protection and rigidity, Sarika, good. Abhishek, Saundarya, Aryan, good. So the cell wall mainly provides definite shape to a plant cell. Also it protects plant cells. So the cell wall is dead. Cell wall is a dead layer which surrounds the plant cell, made up of three structures, that is primary wall, secondary wall and middle lamellae. Cell wall is made up of cellulose, pectin. Very good, Srujana. The cell wall surrounds the plasma membrane of plant cells and provides tensile strength and protection against mechanical and osmotic stress. Good. Priya, good. So, the cell wall is mainly composed of cellulose, hemicellulose, lignin, pectin, suberin, etc. The primary cell wall diminishes as the cell matures. Then inner to primary, we see the deposition of secondary wall. So middle lamella is made up of calcium pectate. Middle lamella is made up of calcium pectate, which helps to keep the cells together, which helps to keep the neighbor cells together. That is the main function of middle lamella. ಇವಾಗ fruit detach ಆಗುತ್ತೆ tree ಇಂದ due to the dissolution of middle lamella. So the middle lamella is made up of calcium pectate, which helps to keep cells together, neighbor cells together. So that is about cell wall. Next we'll move with plasma membrane or cell membrane. S. What's the function of cell membrane? S. What's the function of cell membrane? It is the outermost layer in animal cell. It is bilayer. It is a bimolecular phospholipid layer. Two layer of phospholipids we see. Bilayer of phospholipids. It's a proteins which allow the transport of we have intrinsic and extrinsic proteins in cell membrane which allows the transport of substances. Abhishek gives structure and protects the inner parts. Aryan, only selective substances to pass through it as it is made up of phospholipid. Sarika, it allows passage of selective substances such as water and molecules. S, good. Amsashri, good. So, cell membrane, in plant cell, next to the cell wall, we see cell membrane. Whereas in animal cell, it is the outermost layer of cell and it is not rigid and it is living. Understood? Whereas cell wall is dead. This bilayer, it is made up of two layers of phospholipids, which is traversed by intrinsic and extrinsic proteins that helps in transport of substances. And this cell membrane is selectively permeable in plant cell and it is semi-permeable in animal cell. Cell membrane is selectively permeable in plant cell. That means it allows certain substances pass through it. It is not dependent upon any size of a solute or substances. Whereas in animal cell, it is a in animals it is a semi-permeable membrane. So it allows some substances to pass through it, mainly depend on size of a substances. So it is selectively permeable in plant cell and semi-permeable in animal cell. Main function of cell membrane means it allows movement of substances either by diffusion. Diffusion means movement of substances or solutes from higher to lower concentration. So there is no need of energy. ATP molecules are not required in diffusion process. Whereas some substances, when they move from their lower to higher concentration, they require energy like ATP molecules. So that is called active transport. And also water moves by a process called osmosis. So that is about cell membrane. So next to the cell membrane, we have mitochondria. They are membrane bound organelles. Outer membrane and inner membrane. So inner membrane contains F naught F1 particles. This is called crystal. Priya, good. Mitochondria are called powerhouse of the cell. Why they are called powerhouse of the cell? Mitochondria are seen in both plant and in animal cells. Yes, Amsashri, they produce ATP molecules. For that reason, mitochondria are considered as powerhouse of the cell. So, it is made up of outer membrane and inner membrane. Inner membrane show foldings called cristae. In the cristae, we see F0 or F1 particles or oxysomes or Racus particles. Sumu, good. Srishti, good. They provide energy to the cell. So these Racus or particles or oxysomes or F0, F1 particles contain enzymes. That enzymes are mainly responsible for oxidation of respiratory substrate like C6H12O6, that is glucose in the presence of oxygen to give rise to carbon dioxide and water with a release of ATP. So as it releases ATP, these organelles are called powerhouse of the cells. Understood? So mitochondria having outer membrane and inner membrane. Inner membrane shows foldings called cristae. In that cristae we see F0 and F1 particles which contain enzymes for oxidation of respiratory substrate, where we see the release of ATP molecules. So that is about mitochondria. It is the name mitochondria is given by Brenda. So the next organelle is chloroplast. So this chloroplast, again mitochondria and chloroplast is considered as semi-autonomous organelles. Mitochondria and chloroplast are considered as semi-autonomous organelles because they have their own DNA. Why they are called semi-autonomous organelles? Because of presence of own DNA, they are called semi-autonomous. Only we have two semi-autonomous organelles, that is mitochondria and chloroplast. So the chloroplast, again they have outer membrane and inner membrane. They are also called plastids, inner membrane. So in this inside we see the grana thylakoids. The arrangement of thylakoids are called grana. So this is stroma. So in grana we see the light reaction. Photosynthesis occurs in the presence of sunlight, that is seen in grana. Whereas in stroma we see dark reactions, that is the photosynthesis occurs in the absence of sunlight, that is seen in stroma. So grana is responsible for light reactions and stroma is responsible for dark reactions. So plastids again are of three types: leucoplasts, chloroplasts and chromoplasts. Leucoplasts are colorless plastids. Chloroplasts contain chlorophyll pigment, mainly helps in photosynthesis. And chromoplasts, which contain coloring pigments responsible for different colors which we see in flowers and fruits. Kirtana is grana seen in chloroplast. So these chloroplasts or plastids are mainly seen in plant cells. They are absent in animal cells. Due to the presence of these organelles in plant cells, plants are autotrophic in nature. They are able to synthesize their own food. So leucoplasts are colorless plastids. Chloroplasts contain chlorophyll pigment responsible for photosynthesis seen in leaves, and chromoplasts contain many coloring pigments responsible for different colors and fruits in plants. Then coming to the endomembrane system. So under endomembrane system, we have endoplasmic reticulum, Golgi apparatus, vacuoles and lysosomes. So all these four organelles come under endomembrane system. They are considered as endomembrane system because their functions are coordinated to each other. As the functions are coordinated, these organelles are come under endomembrane system. So endoplasmic reticulum. So this endoplasmic reticulum are of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. See here, endoplasmic reticulum, they extend from outer membrane of the membrane of nucleus. This endoplasmic reticulum, if they have ribosomes, they are called rough endoplasmic reticulum. If ribosomes are absent, they are called as smooth endoplasmic reticulum. So, you all know that ribosomes mainly responsible for synthesis of proteins. Protein factories of cells are nothing but ribosomes. So, if the endoplasmic reticulum have ribosomes, so the main function of endoplasmic reticulum is synthesis of protein. If ribosomes are absent, that endoplasmic reticulum is called smooth endoplasmic reticulum, mainly involved in lipid synthesis of lipid is seen in smooth endoplasmic reticulum. Rough endoplasmic reticulum mainly helps in protein synthesis. The next endomembrane system include Golgi apparatus. See, we have Golgi body. The main function of Camellia Golgi gave the name to this Golgi apparatus to this organelle. The main function of this Golgi apparatus is packaging of substances and released into the intracellular space or out of the cell, and also helps in lipid synthesis. Then coming to the lysosomes. Lysosomes, they contain hydrolytic enzymes. Lysosomes contain hydrolytic. Hydro means water. Lytic means break. So they require water in the breakage of substances like carbohydrates, proteins and lipids. So lysosomes mainly contain hydrolytic enzymes like proteases, carbohydrates, lipases that helps in digestion of carbohydrates, proteins and lipids. It's the main function of lysosomes. And also this engulf foreign substances and thereby they provide immunity to our body, and also it kills worn-out cells or worn-out, yes, they are called suicidal bags of cells. Lysosomes are called suicidal bags of cell because it engulfs the worn-out or damaged cell in our body. That's why they are called suicidal bags of a cell. Then coming to the vacuoles. Vacuoles are larger in plant cells, whereas in animal cell we see smaller vacuoles. So the vacuoles in plant cell mainly helps in osmoregulation. The outer layer of the vacuole is called tonoplast and it mainly helps in osmoregulation in plants. It's about endomembrane system. So the next we have nucleus. See, in eukaryotic cells, cytoplasm is divided. It is compartmentalization of cytoplasm is seen in eukaryotes by the different cellular organelles. Inside the cell membrane, we see cytoplasm, which is divided into many compartments based due to the presence of different cellular organelles. So nucleus, we know that what's the main function of nucleus? In prokaryotes, it is not covered by nuclear envelope, whereas in eukaryotes, it is covered by nuclear membrane. And we see nuclear pores. This is nuclear membrane. Robert Brown discovered nucleus. Nucleoplasm, cytoplasm present inside the nucleus is called nucleoplasm. Inside that we see a circular structure called nucleolus. Nucleolus or nucleoid. So this nucleolus contains genetic material. What's the function of genetic material? Anyone? So this genetic material which is present in the form of DNA, this thread-like structure that condenses to form a chromosome, during cell division. Yes, Abhishek, good. DNA. So DNA, which is present in thread-like structure inside the nucleus, will condense to form a structure called chromosome during cell division. So, Sarika, as nucleus contain DNA, it transmits hereditary character. Yes, hereditary character means it mainly helps to transfer parental characters to their offspring. Kirtana, good, DNA. So, based on the position of centromere, these chromosomes are of different types: metacentric, submetacentric, telocentric, and acrocentric. So, submetacentric means centrosome is present at the center and two equal arms we see in chromosomes. Submetacentric means centrosome is little bit shifted. So, this is metacentric chromosome. If it is shifted little bit above the center, then it is called submetacentric. And in acrocentric, we see one large arm, one large and one short arm. So that is centrosome is towards the end. And in telocentric, only one large arm is seen. That is seen in telocentric. Yes, Srujana. Good. DNA contains only 4 bases called A, T, C and G. A means adenine, T means thymine, C cytosine and G guanine. The sequence of the nucleotides along the backbone encodes genetic information. Yes, very good, Sujana. So, based on the position of centromere, we have different types of chromosomes: metacentric, submetacentric, acrocentric and telocentric, and these chromosomes are responsible for inheritance. Inheritance means carrying parental characters to their offsprings. So next we have centrioles. Centrosomes give rise to centrioles. We have here centrioles, two perpendicular. Centrioles mainly helps in cell division by forming spindle fibers. These centrioles, the centrosomes are seen in animal cell, mainly helps in cell division by forming spindle fibers. And cytoskeletal structure. Filamentous proteinaceous structures form a cytoskeleton. Like inside the cytoplasm, we see a filamentous proteinaceous structures collectively called as cytoskeletal structure, which provides strength, mechanical strength to the cell, and also it gives definite shape to cell and helps in movement of substances inside the cell. So that is about some functional and structural point of cellular organelles present in cell. In detail, we study in our regular classes both plant cell and also animal cells. So we'll see the now slides related to this discussion. See here, we have prokaryotic cell and eukaryotic cell. So as I told, prokaryotic cell is having nucleoli, which is not surrounded by nuclear membrane. Flagella and cilia are seen in prokaryotic cells. So outer layer is cell envelope. Inner to cell envelope, it is cell wall. Then we see cell membrane. So this eukaryotic cell having different cellular organelles. Ribosomes, there are 70s ribosomes seen in prokaryotes and 80s is seen in eukaryotes. See, this is structure of plant cell. Outermost organelle is cell wall. Inner to that we have cell membrane. Larger central vacuole is present. Chloroplast is present, which mainly helps in photosynthesis. Mitochondria. Then cytoskeleton, which is made up of microfilaments, microtubules are seen here, and with a nucleus having rough and smooth endoplasmic reticulum. This is anatomy of an animal cell. Here the cell wall is absent, but here we see cell membrane, nucleus, mitochondria, Golgi apparatus are seen in animal cell. So the next, plasma membrane. See here, plasma membrane, we have outer membrane and inner membrane by layer of phospholipid with extrinsic and intrinsic proteins. Main function is it allows movement of substances and it acts as a selectively permeable in plant cell and semi-permeable in animal cells. Solutes will move either by diffusion or with an active transport which needs ATP, and also water moves by a process called osmosis. So Amsashri, after the bridge course, regular classes will start. The management will decide and tell you the date when our regular classes are going to start. So this is the cell wall which is seen in plant cell, which is dead, having three structures, that is primary wall, secondary wall and plasma membrane. So as I told, primary wall diminishes as the cell matures. Inner to primary wall, deposition of lignin or pectin is going to start. That forms secondary wall, and middle lamella helps to hold neighboring cells together, which is made up of calcium pectate. So endomembrane system. This endomembrane system is composed of different membranes that are suspended in the cytoplasm within a eukaryotic cell. These membranes divide the cell into functional and structural compartments or organelles. So it consists of endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles and vesicles. So we know that endoplasmic reticulum mainly helps in protein synthesis and also lipid synthesis. Golgi apparatus helps in the packaging of substances and transport that into intracellular compartment or outside the cell. Lysosomes are suicidal bats. They engulf worn-out cells and also they kill foreign microbes which enter into our body. Vacuoles are larger in plant cells and very small in animal cells. These vacuoles in plant cell mainly helps in osmoregulation. Mitochondria, it is semi-autonomous organelle seen in both plant cell and in animal cell, having outer membrane and inner membrane. So inner membrane folded into enfoldings of inner membrane is called cristae, where we see the oxysomes or rachous particles which contain enzymes for aerobic respiration. So that helps in the oxidation of respiratory substrate like glucose to release carbon dioxide and water with ATP molecules. That's why mitochondria are called powerhouse of the cells. Plastids. So again, plastids are made up of outer membrane and inner membrane. So again, the arrangement of thylakoids inside this membrane is called granum, where we see the light reactions, and in stroma we see dark reactions. These plastids are seen only in plants responsible for photosynthesis. That's why plants are autotrophic in nature. Then ribosomes. They are membraneless organelles seen in both prokaryotes and also in eukaryotes, mainly responsible for protein synthesis. They are made up of two units, one large subunit and one small subunits. So 70S ribosomes are seen in prokaryotes and 80S ribosomes are seen in eukaryotes. So if the endoplasmic reticulum have ribosomes on their surface, they are called rough endoplasmic reticulum, mainly helps in protein synthesis. And if these ribosomes are absent, then that type of endoplasmic reticulum is called smooth endoplasmic reticulum or agranular reticulum. That helps in lipid synthesis. This is about ribosome. Nucleus. It's a very important organelle which controls functions of cell. So it is having nuclear membrane or nuclear envelope. Inside that we see a cytoplasm that's called nucleoplasm, a nucleolus, a nuclear pore and chromatin. So these chromatin, we have again heterochromatin and euchromatin. So these chromatin condense to form a chromosome during cell division. So that, see here how this chromatin is condensed to form chromosome. You can see clearly in this diagram how the DNA is, which is present in thread-like structure, condensed to form a chromosome during cell division. The structure of chromosome we can study only in cell division, that too in metaphase of cell division. So types of chromosomes based on the position of centromere or centrosome. So here see metacentric, where the centromere is present in the center of chromosome, and submetacentric, it is centromere is shifted little bit above the middle, that is submetacentric. In that we see one large arm and one short arm. Whereas in acrocentric, the centromere is shifted towards the end of chromosome, where we see very small short arms and long arms of chromosomes. Whereas in telocentric, it is shifted towards the end and we see only two long arms. There is no short arms in telocentric. So these types of chromosome based on centromere. So thank you. So this is about the cell, the unit of life which performs definite functions. So the cell contain different different cellular organelles with different structures and functions, so that all the functions of the cellular organelles coordinate to each other with the help of nucleus. So we study in detail about these cellular organelles, their structure and function in our regular classes. Okay, thank you one and all.